If you have spent any time in recovery, sports medicine, or peptide research communities, you have almost certainly heard of TB-500. It gets described as the full-body recovery peptide, the systemic healer, the compound people reach for when the injury is not just one thing, when the whole system feels like it is running behind. It is frequently paired with BPC-157 in what the biohacking community calls the Wolverine stack, and it has been used by athletes, clinicians, and researchers across multiple countries for years.
It has also just come through the most consequential regulatory moment in its history. On July 23, 2026, this week, the FDA's Pharmacy Compounding Advisory Committee voted on whether TB-500 should be recommended for inclusion on the 503A bulk drug substances list. That vote and what it means is something we are going to cover in full detail. Let's start with the name, because the naming around TB-500 is where most explanations begin to go wrong. TB-500 is not thymosin beta-4. Those two things are related, but they are not the same. Thymosin beta-4, abbreviated TB4, is a naturally occurring protein. It is one of the most abundant proteins in the human body. It is found in essentially every cell that has to repair itself: skin, cardiac muscle, tendon, cornea, neural tissue. It has been circulating in your bloodstream and accumulating at wound sites your entire life. That distinction matters for a specific reason. The evidence base for thymosin beta-4 is considerably larger than the evidence base for TB-500 specifically. When someone cites a study on thymosin beta-4 to support a claim about TB-500, they are making an assumption that the 7 amino acid fragment produces the same downstream biology in human tissue as the full 43 amino acid protein. That assumption may be reasonable. It is not proven. And as we will see, the FDA's July 2026 review drew that distinction explicitly. To understand what TB-500 is trying to do, you need to understand what thymosin beta-4 actually does in the body, because the natural protein has genuinely well-characterized biology. Thymosin beta-4 is not exotic. It is present in virtually all mammalian cells, and its central biochemical role is in actin regulation. Actin is the cytoskeletal protein cells use to change shape and move. It exists in two forms: filamentous actin, organized into structural strands, and globular actin, the individual unpolymerized units that can be recruited into new filaments when needed. Thymosin beta-4's primary characterized function is sequestering globular actin. It binds to and holds individual actin monomers, regulating how much of them are available to polymerize into new filaments. That single mechanism, actin sequestration, sits underneath most of what the broader thymosin beta-4 literature explores, because actin dynamics are central to one of the most fundamental repair processes in biology: cell migration. When tissue is injured, repair cells have to physically travel to the site of damage before they can do anything useful. They extend projections, grip the surrounding matrix, and pull themselves forward. That movement depends on precise actin polymerization at the cell's leading edge, and thymosin beta-4, by regulating the pool of available actin monomers, is one of the molecules that controls how readily that movement happens. Beyond actin, thymosin beta-4 has been studied for roles in angiogenesis, the formation of new blood vessels, anti-inflammatory signaling, and in cardiac tissue specifically, the activation of dormant progenitor cells after injury. In the heart, thymosin beta-4 activates downstream signaling through AKT phosphorylation, promotes the ILK-Pinch-Parvin complex, and suppresses NF-kappa B, collectively reducing cell death and inflammation after cardiac injury. A 2025 study in cardiovascular research provided some of the first human clinical evidence for this cardiac pathway. Recombinant human thymosin beta-4 showed improvement in ischemic cardiac dysfunction in STEMI patients after reperfusion. That is full-length thymosin beta-4, not TB-500. But it demonstrates that this biological pathway has genuine clinical relevance in humans,
and it is the mechanism that the shorter TB-500 fragment is designed to access. Let's go deeper into the specific mechanism, because this is where TB-500's proposed biology becomes genuinely interesting, and where understanding the detail helps you evaluate the claims you encounter. The 7 amino acids in TB-500 correspond to what researchers call the actin binding domain of thymosin beta-4. That is the specific region of the full protein responsible for sequestering G-actin. When TB-500 binds to a G-actin monomer, it holds that monomer in a sequestered pool, not incorporated into filaments, not doing structural work, but available to be rapidly deployed when a migration or repair signal arrives. Think of it as a reserve system. TB-500 maintains a larger reserve of deployable actin monomers. When the injury signal says, "repair cells needed here," those monomers can be rapidly mobilized into new filaments at the migration front, allowing repair cells to move more quickly and efficiently toward the damaged tissue. That is the core proposed mechanism. Not a hormone, not a growth factor in the conventional sense, a regulator of the cellular machinery that controls how quickly repair cells can move. Now, why does this matter specifically for the tissue types TB-500 is most discussed for? Tendons, ligaments, and muscle tissue share a common repair bottleneck. Repair cells, fibroblasts in connective tissue, satellite cells in muscle, have to migrate into injured areas before collagen deposition, matrix remodeling, and structural repair can begin. The speed and effectiveness of that migration directly influences the timeline and quality of healing. If TB-500 genuinely accelerates the actin dynamics behind that migration in the relevant cell types, and the preclinical evidence suggests it can, the downstream effect is a repair environment where the second phase of healing is initiated more quickly and more fully. One more element worth noting in the mechanism. In addition to actin sequestration, TB-500 has been studied for upregulation of cell surface receptors involved in migration signaling and for anti-inflammatory effects in injured tissue environments. These additional proposed effects expand the mechanism beyond pure cell motility, but they are also the elements with the least mechanistic characterization in the literature. The actin sequestration story is the most solid foundation. Everything built on top of it requires clearer evidence. The animal evidence base for thymosin beta-4, and by extension TB-500, is substantial across multiple tissue types and multiple research groups. That is a meaningful distinction from some other peptides in this space, where the literature is concentrated in one laboratory. Let's go through the major areas. Wound healing has the deepest preclinical base. Animal studies have reported 42 to 61% faster re-epithelialization rates with thymosin beta-4 compared to controls, re-epithelialization being the process of new skin cells covering a wound. That range comes from multiple studies across different wound models, which gives it more weight than a single isolated finding. The skin and wound healing evidence also has the most developed human translation path. Phase 2 clinical trials in venous stasis and pressure ulcers demonstrated accelerated healing by nearly a month compared to placebo. That is full-length thymosin beta-4, not TB-500 specifically, but it represents the strongest clinical evidence in the entire TB-4 literature. For tendon and connective tissue, the application most people discussing TB-500 are interested in, the preclinical signal exists across cell and animal models. Studies show accelerated repair cell migration, improved collagen organization, and faster functional recovery in injured rodent models. The important caveat here is the same one we applied throughout the BPC 157 video. A rodent tendon injury and a human athlete's torn ligament are different in ways that matter. Different tissue architecture, different immune response, different mechanical environment, different scale. The animal evidence is a biological plausibility argument. It is not a clinical efficacy claim. Cardiac tissue is where some of the most scientifically significant findings have emerged. Thymosin beta-4 has been described as the first molecule demonstrated to simultaneously trigger both myocardial and vascular regeneration after systemic administration in animal models. The mechanism? Through AKT signaling, progenitor cell activation, and NF-kappa B suppression, is consistent across multiple studies. And in 2025, the first human clinical data in cardiac tissue was published. Recombinant thymosin beta-4 in STEMI patients showed improvement in ischemic cardiac dysfunction. Again, full-length protein, not the TB-500 fragment, but the human signal exists in cardiac tissue. Neural tissue is the frontier. A 2026 study tested thymosin beta-4-derived peptides in neuroinflammation models and Alzheimer's mouse models, finding alleviation of neuroinflammation and neurite atrophy. This research is very early and very preclinical, but it shows the actin-regulating biology of thymosin beta-4 reaching into neural repair, an unexpected extension of the core mechanism. One finding in the liver literature deserves specific mention because it is a note of caution rather than encouragement. A 2023 study found that targeted deletion of thymosin beta-4 in hepatic stellate cells reduced liver fibrosis in a mouse model. What that suggests is that thymosin beta-4's tissue repair effects are context-dependent, healing in some organs, potentially contributing to fibrosis in others. That context dependence is not a reason to dismiss the compound. It is a reason to be precise about which tissue, which condition, and which evidence you are evaluating. The same filter we apply to every compound on this channel. Let's apply that filter now to the human evidence, because this is where the gap between thymosin beta-4 and TB-500 specifically becomes most important to understand. Human clinical evidence for full-length thymosin beta-4 exists and is meaningful in specific contexts. Phase 2 trials in wound healing, venous stasis ulcers, and pressure ulcers showed statistically significant acceleration of healing. The ophthalmic program, using thymosin beta-4 eye drops for dry eye and neurotrophic keratitis, has reached Phase 3 trials and shown benefit on objective endpoints. The 2025 STEMI cardiac study showed improvement in ischemic dysfunction. These are real human signals for the full protein. For TB-500 specifically, the 7 amino acid synthetic fragment, the picture is different. As of mid-2026, no completed randomized controlled human efficacy trials have been published for TB-500 in musculoskeletal, wound healing, or recovery indications. No published human pharmacokinetic data for TB-500 exists. The doses used by people today are extrapolated from animal studies and clinical experience, not from controlled human pharmacology research. That gap is not a reason to assume TB-500 does not work. The underlying biology is real. The preclinical signals are consistent, and the full-length protein has human evidence in related applications. What it means is that any specific efficacy claim for TB-500 in human musculoskeletal injury, this healed my hamstring, this fixed my shoulder, cannot be verified against controlled trial data. The experience may be genuine. The attribution to TB-500 specifically, at a given dose through a given route, producing a specific outcome in human tissue, is not established by the published evidence. One additional complication. TB-500 has an interesting history in veterinary medicine, specifically in horse racing, where it was used to accelerate injury recovery. Equine doping controls developed detection methods for TB-500, and horse racing authorities banned it in multiple jurisdictions. The equine evidence contributed to human biohacker adoption of the compound. The caveat is that equine pharmacology does not translate directly to human pharmacokinetics. Horses and humans metabolize compounds differently. The dosing protocols that circulate in human use communities rest on equine pharmacology assumptions that have not been validated in humans. On July 23, 2026, the FDA's Pharmacy Compounding Advisory Committee met at the White Oak campus in Silver Spring, Maryland, and voted on a question that has been building since 2023. Should TB-500 be recommended for inclusion on the Section 503A bulk drug substances list? The committee voted 8-6 in favor, with one abstention, to recommend TB-500 for possible inclusion on the 503A compounding list. That is a narrow majority. and several things about that result are worth understanding precisely.
This is an advisory vote. The FDA is not bound by PCAC recommendations. Even a unanimous positive vote does not immediately legalize compounding. What follows is a formal rulemaking process: notice, comment period, FDA review, final determination that typically takes 6 to 12 months. TB-500 cannot be legally compounded by 503A pharmacies today. That will not change overnight as a result of this vote. What the 503A review specifically evaluated was TB-500 for wound healing. The committee reviewed available safety and efficacy data, considered public comments, and voted on whether the evidence was sufficient to support inclusion for that indication. The FDA's own scientific briefing document noted that TB-500 is a 7-amino acid fragment, the LKKT-ETQ sequence, and drew the explicit distinction between that fragment and full-length thymosin beta-4. The agency found no adequate published effectiveness or safety evidence for the fragment specifically in wound healing, which is part of why the vote was as close as 8-6 rather than unanimous. Also at the same meeting, BPC-157 passed 8-6, MOTS-C passed 7-5, KPV passed, Samax and Epidolon passed on day 2. The only compound voted down was DSIP, delta sleep-inducing peptide, which the panel rejected 6-7. 6 of 7 peptides reviewed passed their advisory votes. TB-500 was one of them. The rulemaking clock has started. Compounding pharmacy access, if it comes, is months away, not days.
TB-500 is almost never discussed in isolation. It is almost always discussed alongside BPC-157, the combination the biohacking community calls the Wolverine stack. So let's address that directly. The rationale behind the combination is mechanistically coherent. BPC-157's most characterized effects center on angiogenesis, the growth of new blood vessels, nitric oxide signaling, and barrier tissue protection. TB-500's most characterized effects center on actin sequestration, repair cell migration, and cell motility. In theory, those two mechanisms are complementary. BPC-157 addresses the blood supply problem, making sure the repair environment has adequate oxygen, nutrients, and immune cells. TB-500 addresses the repair cell movement problem, making sure fibroblasts and other structural repair cells can migrate efficiently into the damaged area. The honest caveat is that this mechanistic logic, while coherent, has not been tested in a controlled human trial as a combination. No randomized study has compared BPC-157 alone versus TB-500 alone versus the combination versus placebo in human subjects. The Wolverine stack is a reasonable hypothesis built on complementary preclinical biology. It is not an evidence-supported treatment protocol. Combining compounds also creates the evidence problem we covered in the BPC-157 video. If someone uses both compounds and experiences recovery, attributing the outcome to either compound specifically becomes very difficult. The confounders multiply with every additional variable.
Thymosin beta-4 is real biology. The endogenous protein has well-characterized functions across multiple tissue repair systems, and the evidence for the full protein in wound healing and cardiac applications has reached human clinical trials. TB-500, the synthetic fragment, is a reasonable attempt to access that biology in a more targeted form. The preclinical signals are consistent across multiple labs and tissue types, which is more than can be said for many compounds in this space. The human evidence gap is real. The fragment versus full protein distinction matters, and the regulatory status is in active transition rather than resolved.
When you hear a claim about TB-500, run it through the filter. What tissue? What route? What endpoint? What proof? If those answers are vague, the claim is ahead of the data. Subscribe to What the Health for science that goes all the way down. The Wolverine Stack Deep Dive, BPC-157 and TB-500 together, is next.